Cloning, gene editing, CRISPR. These aren't just buzzwords or fantasy. They're tools now shaping agriculture, medicine, and even conservation. But what does it mean for all of us? In this video essay, we explore where cloning and gene editing stands in 2025, what we've achieved, where we're going, and what's still too dangerous to touch. Let's start with gene editing, precise modification of DNA within living organisms. The star player here is CRISPR-Cas9, a kind of genetic scissors that can cut and rewrite strands of DNA with remarkable accuracy. Since its emergence in the early 2010s, CRISPR has evolved. Now we also have basic editing and prime editing, which allow even more refined tweaks, like changing a single letter in a DNA code without making a full cut. Sounds small, it's not. In 2020, the first patient was treated in the body using CRISPR to tackle a rare eye disease. In 2023, the FDA approved a CRISPR-based therapy for sickle cell disease. So we've gone from lab bench to the hospital, and fast. But the line between treatment and enhancement is thin. In 2018, a Chinese scientist stunned the world by announcing the birth of the first gene-edited babies. He claimed to have altered their DNA to make them resistant to HIV. The global response? Outrage. That moment, now referred to as a major ethical breach, triggered bans, crackdowns, and calls for global consensus. Why? Because editing germline cells, which are sperm, eggs, embryos, means those changes can pass on to future generations. It's irreversible, and we're not ready yet, or so it seems. Today, most countries allow somatic editing, treating diseases in existing people, but strictly prohibit germline manipulation. The science may be ready, society not so much. Let's shift to cloning, the older sibling of gene editing. The first breakthrough came in 1996 with Dolly the sheep, cloned from an adult cell. It was a milestone, and it sparked fears of human cloning. Nearly 30 years later, here's where things stand. Animal cloning is routine, especially in agriculture. In the pet industry, companies offer cloning services for your dog or cat for the right price. And in conservation, scientists have cloned endangered species, but human cloning remains off limits. No verified attempts have ever been successful or ethical. International law bans reproductive cloning in humans. Therapeutic cloning, creating embryos to harvest stem cells, is also controversial and rarely practiced. In agriculture, CRISPR is revolutionizing crops. Tomatoes with longer shelf life, corn resistant to drought, rice immune to devastating fungi. These are real, and many are already on the market. In livestock, gene-edited pigs are resistant to diseases which cost farmers billions each year. And beyond that, some projects are venturing into de-extinction, trying to revive woolly mammoths or the dodo bird using gene editing and cloning hybrid techniques. It's science fiction inching towards science fact. We can think of Jurassic Park. Would you go on that ride? So we've made huge strides, but here's a twist. The science is moving faster than our rules, and far faster than our ethics. Who gets to decide what disease is worth editing, and what's just difference? What happens when gene editing becomes a luxury only the rich can afford? And what about unintended consequences, off-target effects, long-term generational risks? Even with strong regulations, rogue actors, private labs, and geopolitical pressures could easily lead to misuse. We're building the future, but are we ready to live in it? Are we ready to live in a world of just seedless grapes? Cloning and gene editing offer power to heal, to restore, even to reshape evolution. But with great power, cliche or not, comes great responsibility. The question is no longer if we can do these things. It's whether, should we, and how. The future is now editable, but it still needs a human touch. Thanks for watching. If you found this video insightful, leave a comment below. Should we ever edit human embryos?